Hello friends, I welcome all of you to Mannan's e-learning program. This is a technical lecture, and in today's lecture I am going to talk about do's and don'ts for a new lab personnel. This is a story of Ryan. Ryan is a new PhD student who has recently joined a very reputed laboratory, and he wants to be before he's kind of nervous also because he's never been to a international lab, and he wants to know what are the ethics which has to be followed in a lab. So like Ryan, the story could be for you. Maybe you are a BSc student, or you are an MSc or MTech or a PhD student, and you are interested in knowing that what should be the decorum one has to maintain, similar to the Ryan story. So let's begin here. So you can see here, this is a very beautiful lab which has been shown. In a lab, this is a typical working bench. As you can see, if you can see here in my arrow, you get lab stools, you get certain equipments, like for example there is a centrifuge machine, as you can see here. Then you can see here is your gel documentation probably system. You also have a microwave oven, as you can see here. A very beautiful view which is very also important. The racks which you can see here with certain chemicals. Okay, you also see a rockers, a gel documentation gel pack, okay, where you can run your gel and so on. So you will be in today's lecture will be get acquainted with this, that what is a lab looks like, what one has to follow in the labs, what are the do's and what are the don'ts. Okay, so in a particular lab you usually come across with a lab in charge. Okay, the lab in charge could be the principal investigator. Usually it is a professor, or it could be an early career principal investigator as well. Besides this, you will also find lab manager, a lab manager who is responsible for maintaining the lab records, lab inventory records, and a day-to-day functioning. And you find postdocs in a lab. Okay, so the postdocs is, like you can say, one of the backbones of a research labs. Besides this, you will find the graduates or the PhD students in the laboratory. You will also find out certain undergraduate students in a lab. Now this undergraduate in Indian scenario, we usually consider to this as a BSc or BTech students. Okay, and the graduate, we call them in United States is the person who is doing a PhD. Okay, but doesn't matter, we are just trying to find out that what is the instructions one should follow, what is the decorum one should maintain, how one should get familiarities with a lab. Just sitting in your home you can get benefited. So the first thing which you should be aware about is the lab space and the lab environment. Now typically a lab bench which will be allocated to you will be like this. You will be allocated with a benchtop centrifuge machine. You will be provided with the racks. You will get the pipette set of your own. Then you will also get a table where you can put certain accessories over there. So that is how, which is will be your working bench. Usually a lab has a separate study rooms that will be in a separate room, or if it is not available, then one can always visit the library or to another classrooms to do his or her data analysis part, or want to read something, so that can be done. It is not like that you cannot do that it in a laboratory condition. Yes, you can do in a lab also the reading part as well. Like when I was doing my PhD, and by the way, those who do not know me, I did my PhD from IMTech Chandigarh, which is one of a premier CSR laboratories. So I've been also associate given a lab space like this. And then besides this lab bench, you should be familiar with the washing area. As you can see, this is a washing area, and then shown is the racks which you will find here. In some laboratories who have the lab technicians, they make things very easy for you because they can prepare certain things in bulk, and they can also provide you certain buffers or the stock solutions which is present in the lab. But in some other laboratories, one has to do the self reagent preparation. So that's also okay. It depends upon the lab to lab variations. Okay. Besides this, in a laboratory condition, you should be familiar with the safety of yourself, okay, and the safety of your fellow workers. So the safety, like you should look for where is the fire extinguisher. If you do not know how to operate it, so usually it is written that how to operate it. So get familiar with the fire extinguisher. Small cuts and burns happens or might happen, so you should be familiar with the first aid box, where it is been located. So you should look into that in a laboratory. If you're not, then you can ask some senior members or maybe the lab in charge itself that where these materials are. You will also find the pipette set, as I initially mentioned, or certain pipettes, like you might get a pressure papers depending upon the lab. You should be familiar with the certain safety equipments like the lab coats. You should always wear a lab coats. You should wear a hand gloves when you are dealing with certain carcinogens, okay, or a mutagenic agents. You should use a protective shield. In fact, some places you have to wear mask even for weighing certain chemicals. So you just look into those things. Each laboratories, they make this inventory. By the way, what I am talking about, a lab which is a cell biology or a microbiology or a molecular biology lab, so all these things is more or less is common over there. Besides this, you should be very much, it is very important that where is the dustbins, and these dustbins not only within the lab but as well as the bulk disposables of the lab. Each lab usually has a lab disposable registry, and it is discarded on a daily basis, and a person who basically comes and pick up. So you should be familiar with which types of box has to be used, which is a general purpose dustbins and which is not a the radioactive hazards dustbins. And usually for E6 they made it color code like red, green, blue, blacks kind of a thing. So get the first thing which you should be, as you enter into the lab, when you have been allocated a lab space, then you get familiarity with the things, and like the safety familiarity, your bench, where is the washing area, where is the autoclave area, what are the instruments which is present in the lab conditions. Okay, so that is the first step. The second steps is the instruments or the tools which you are going to handle. Like in some universities or in some research laboratories, they have central instrumentation facilities, okay, or a common room facilities which is there. In this facility is been provided, and like 2 or 3 labs or maybe a whole floor lab might be using this, because each and every scientist cannot afford all the chemicals. Some of these chemicals is in crores or in lakhs of rupees, so they make it a common facility, so it is available over there. But again, so one has to be very where is a common facility if something which is not present in a laboratory condition. So get familiar with this. Then there is usually one has to make an inventory, or one has to make a lab register. For that you should make a proper entry so that the lab in charge of that central instrumentation facility or a common facility should not pinpoint to you. Like you will also come across, these are few general things I put, but this may, depending upon the laboratory or the labs, this may differ. Like this is a microscope which has been shown. This is a compound microscopes. You will get a glasswares. You might be need a separate rooms for your laminar hood, depending upon the labs, whether it is a BSL 1, 2 or 3 labs, then they all have their different facility. I'm just restricting myself to BSL 1 and BSL 2 lab facilities, and in a common general labs what are the tools which is there. After this, one should be very much familiar with the MSDS. Okay, MSDS stands for Material Safety Data Sheets. Each laboratory has, as I said in the very beginning, that a lab manager makes the inventory, or if the lab manager is not there, then the principal investigator or lab in charge is all familiar with this. Now, why this is very much important? Because when I say we, means when we go into the laboratory and for research purpose, then we actually deal with certain instruments. We also deals with certain chemicals. Some of these chemicals could be toxic. Okay, so like the toxic sign, some of the chemicals could be a biohazard, or some of the chemicals could be a radioactive hazards. Some chemicals could be an irritant. Like if you are dealing, just for simplicity sake, like say for example you are working in a hydrochloric acid. Now this hydrochloric acid is a kind of a toxic for this, or maybe someone who is even a simple SDS, that is the sodium dodecyl sulfate, you are going and weighing this, then you should know that it can cause a irritant to you. If you are handling and working with ethidium bromide, like for your gel purposes, then this ethidium bromide is a potential carcinogen. So you should be very much, whatever the chemicals which you are going to open for weighing purpose, for making the reagent purpose, you should read and get familiarity with this. For ease sake, most of the bottles, whether it is a room temperature chemical or a 4-degree reagent, or some of the things which might be taking from -20s also, like if it has been stored there for certain stock solutions, you should be very much familiar, and accordingly a safety measures can be taken, like for your own protection. And sometimes due to some accident, like there is a spillage, then how to get rid of that spillage. Like for example, if someone who is working on a radioactive materials, then there will be a separate room where to take that radioactivity handling, how to discard the liquid radioactive waste, how to discard the solid radioactive waste. That familiarity one should be taken care. If you're not dealing with that, even for like if you're dealing with a microorganisms or a biohazard organisms, then there is one should be very familiar with these things, that what could be a potential risk involved in the process. Okay, so that is the first thing. Like after the lab environments, lab equipments, common facility, getting the know-how, one should look for the material safety data sheets. Then there are certain things which I have tried to written down here, is like one should be familiar as the location and proper use of fire extinguisher, because these things happens. I do not want to name a university, because this is one of my personal experience. This happened in one of the university, that one of the student was working and then there was a fire which has been caused, and this fire might cause, like if you're handling spirits or you are handling alcohols, they are very inflammable. So one should able to know where is the fire extinguishers over there. Maybe if you're handling with say phenol chloroform, and if you somehow forgot to wear your gloves, then it might spill over there. Then one has to wash your hands and then look for certain burn creams and then apply over there. So that is the first thing. After getting used to this thing, where this is available, where I can easily locate them. Okay, so that is one of the do's. Then if there is any kind of injuries happen, like somebody might, because of certain chemicals, like for example formaldehyde, if somebody is using, there is a chances that person, if they have not used a mask or not used a chemical hood, then it might be inhaled. There are certain chemicals which can inhale and can cause headache. So that type of symptoms if somebody is having, or there is a spillage which occurs, be confident, because these things happens. Okay, like one of the most common thing is the breakage of the glass, which like test tubes or petri plates, it happens. Okay, so just make an inventory of that and report to the lab manager or lab your in charge, that see, this happens with me, I broke this glass pipette, or maybe I broke this petri plates. That's fine, everybody knows about this thing. Or if you have inhaled something, so a first aid can be given to the person. Or maybe if it is very severe case, then in a university or in research laboratory, mostly they have hospitals or nearby hospitals, one can be taken care. So don't afraid of saying those things, don't hide things. If something happens, you just tell the person who is responsible for doing who is taking care of the laboratory. So tell to the instructor. If you are also working in a say classrooms also, if something happens, there is no way, because to hide those things, tell, be confident, tell, see, this happened and it's broke with me, this is perfectly all right, rather than hiding and having a guilt with you. Okay, use protective mask. That's what I was also showing in that there is a protective glass shield which is used. Like for example, if you're using or visualizing your gel, then you should use a UV protective shield over there, or UV protected goggles to visualize your DNA. Or you maybe you are want to wear a mask over there, because certain chemicals may be inhaled from your nostrils or from your mouth, then one should use a protective mask for there. Okay, then gloves. Like if you, as I said, most of these discussions which I said is for the cell biology or molecular biology laboratories. So if you're setting is a simple experiment like PCR or a polymerase chain reaction, or you are handling your gels, always wear gloves, because some of the things is potentially irritant or can carcinogenic. Or one of the most important thing is like our hands. These hands has got some proteases, DNases and RNA. So whatever the biomolecules you are working upon, that might be degraded if you do not wear the gloves. So wear gloves wherever it is necessary for handling your things. Then when something you are taking which is hot, because maybe you're using a microwave oven to make, like say for example, a gross gel preparation, or maybe you are taking certain things from your autoclave, then there is a different types of insulated gloves which is available, every laboratory has got. So if you getting things from after autoclave, after your media preparation and reagent preparation, they are very hot. Okay, like 121 degrees Celsius, maybe you bring it to 100 degree, but still it is too much temperature is there. So don't use a normal gloves for this, you should always wear a insulated gloves. Even this is also necessary like when you are opening a minus 80 degrees, because a cold burn can also occurs. Or if you are doing a biochemistry experiment, then you should use tongs over there, or you should use certain tissue paper. Even the tongs is not available, you can make a simple tong and then you can hold over there. But mostly most labs has got tongs to hold the pipettes, or you can use them like taking out certain things from water bath or things, then one should able to wear that. Okay, then when somebody is working in hot plates, okay, and water bath, because in say DNA extraction, maybe you are using a heat thaw method, and then you are using a temperature of say 100 degrees or boiling temperature, so then that becomes very much imperative. Then some students do these mistakes very often. I used to do it. Is not like when I was student I didn't did that. there is no point of mask hiding these things. That is label your things very properly. I will show you a picture that what I meant over there. Proper label should be with a date, okay, with your specimen number, and for whatever the initials which you have or the initial of your lab in charge should be there when you are using a central facility, as I said, or a common room. Because sometimes if you are growing certain bacteria or fungus in a incubator shaker and you didn't label it, now in a common facility you're doing it, maybe the lab in charge will remove your culture and say some bacteria might takes a week time to grow, or you are waiting for a week time to grow and you didn't label it. So after a week time, somebody else might take your take your media component or take your flask with them. So proper labeling is very important for the not only the media, but as well as it is very necessary for your small microfiche tubes which you are using. Okay, then always follow the instructions of the lab manager or the manager or the instructor who is ever telling you that a proper disposable, as and I showed you the picture in a very beginning. So whenever you finish your work, say suppose you did a whole day work, then don't keep your things as such on your table. You wash them, keep in the washing rack, clean the area over there. Like let me give you an example, like the weighing area, which is most commonly used for say reagent preparation or media preparation. If you are not cleaning that area, then one is actually adding or calling the microbes to grow over there, and then there will be contamination. So proper cleaning is very much important. So these are some of the do's or the decorum one has to follow in the laboratory. It is a generalized description which I just said, and it will depending upon the lab you are working upon, and based on each lab they have specific sets of protocols, so follow that. Okay, don'ts. The don't which I mentioned here is that do not discard biological waste in a normal dustbin. There are normal dustbin for routine purpose, like you have tissues, you might have certain gloves which you can, just simple gloves which you are not using a biological materials, that is you using in a normal dustbin, or maybe paper dustbins which are using it. So don't throw it. Any biological material which you are handling, okay, there is a chances of potential hazards, and this all comes by the way in a good lab practices, which is we called as good lab practices. So it has to be autoclaved. Even say you didn't get your results, maybe it is a contamination, but still you cannot keep as such over there. Means you cannot keep keeping lying on your table or a fridge. You should after that it is your responsibility that you take out your biological samples and autoclave and then discard. What I have shown here in this particular picture, this is specifically from the Punjab Pollution Control Board, and this will differ from again from different states and different countries as well. But as what is essentially what you can see here, that there is a red color plastic container, let me change color code, or a yellow color plastic container or a blue color plastic container, and they are written here that what type of material one has to be put into. So there is a specific segregation of infected as well as non-infected waste, and if you are not familiar with this, each laboratory has a waste disposable systems. So as I said in the very beginning, make sure where is this waste disposable is there, make a entry of this. Next time when you visit to a hospital, just see the hospital setting. They probably have these types of settings over there. Sometimes we don't look into this because whenever we visit hospital is because of the some ailment, or we visit someone who is admitted over there and our whole focus is over there. We just met the doctors or the nurses or maybe the chemist, but we never look into these things which has been there. So every hospital has a weight segregation system. So next time whenever you visit it, you kindly get noted these things. But if you are a lab person like a Royan, which I said, you will be get familiar with these things, and before starting an experiment, the first 1 day or 2 days you should get familiarized with these things. So do not waste, as I repeat, biological waste materials. Like for example, even the ethidium bromide, the gels, there is a specific disposable that it has to be disposed into a particular blue color container, or maybe the labs have separate segregation based containers which is present. Discard over there. Okay. Then for the personal don'ts, one should not use, don't wear chappals, okay, or you can say any shoes which is where your fingers is or your feet is been exposed. So these type of things is no, you cannot wear these things. You should use, for girls specifically, it should be a tapered end, or at least it should be tapered end. The best would be like you take the whole shoes and it should be covered. In some cell biology or some microbiology laboratories, they might have their own shoes and they tell you to keep your shoes outside and then wear that. So then wear that thing. So normal shoes, if it is leather shoes, that will be preferred, but a fabric shoes can be used depending upon which labs you are, but at least your toes should be covered. Okay, that is the one thing. Another thing which one should not do is that you should not keep your hairs loose. Okay, you should tie your hairs, means it should be properly been closed. For me it is not applicable because I don't have hairs, but one has to properly tie their hairs. Okay. Like for example, if you are using a Malditoff, one of the scientists they considered, and this is my personal story I'm sharing here, that they found that in their Maldi they found one of the protein which is actually coinciding with the sheep protein. Later on they find out that it is the sheep proteins which they are finding. By the way, they were working on the human cell human DNA. That sheep, then they isolated the proteins, and then they humans fibroblast cells, and then they isolated the protein, and then doing the Malditoff. Then they found out that this protein is from the sheep. Now why that occur? And then later on it was found out that it was the sweater which someone was wearing, this because it was a winter, and from there that sweater it's actually gets contaminated. So you can imagine that if something is loosed open, or if you're not wearing a lab coat, what kind of your experiments can turn out to be. So don't do that thing. Labels, as I was mentioning, this is, as you can see, label here mentioning 1 2 3 4. For a particular set of experiments, if you're labeling in 1 to 6, like example for a test tube, that's fine, but you make a lab notebook and mention that what this 1 refer to, what is 2, this 4 and 5 for the working part. But when you're storing something, then you should properly label with the specimen name, your date and some initial marks, so that you can always find out what it is there. That's why not only the labels, but a proper lab record keeping is very much important over there. And some students or some technicians, they be careless with that. Contamination in refrigerator. If you are keeping or you are working on a bacteria or a fungi or something, maybe after, even if you are keeping at 4 degrees Celsius or so, there could be certain cross contaminations occurs. This is very usual. So try to keep your refrigerator, don't like your work has been done, then it is hanging for a month or so in the refrigerator. Take that samples. If it is contaminated, if you can revive it, if you cannot revive it, just discard it by autoclaving. So that is again very much important, that don't do it. Spillovers and cleaning, I mentioned it earlier as well, that before leaving a lab, clean your desk area, clean the utensils. In terms of, when I say utensils, is the tools which you are using, like test tubes or a petri plates or whatever materials which you are using, clean and put in a proper place. Another thing, don'ts is no eatables. When we say no eatables, no food or drink, even water is not allowed in a laboratory condition. If you want to take a sip of water, go outside the lab and take it, or maybe you go to a study room which is usually associated or in a adjacent area, then take your eatables over there. Or never ever do that thing in a laboratory condition. Lab records, this is one of major thing which actually students or the lab technician or the lab personnel forgets, or a researcher forgets, that they don't update their record. So update your record in your lab manual or in your lab notebook. Keep your, as you can see here, it is such a beautiful picture which has been shown, that this particular person has wrote everything, put the gel picture over there, looks like it is a protein gel or maybe autoradiograph gel, and see properly labeled it, what is the observation, and making a record keeping. So once you performed an experiment, okay, once you have performed your experiments, and after observation, you should note it down in your lab record, okay, and update it. Don't say that okay, I will analyze later on. Even if it is a negative result, or if it is not the expected result, but still note it down. You never know. That's the beauty of the science. Something novel might come, or a serendipity might occur. Like we know the discovery of the penicillin, that it is something which Alexander Fleming was not working upon, but he observed something else, and then it leads to a discovery of the penicillin. This lab record and lab keeping is also very important if you are going to file your patent. Whenever you file a patent officer, if there is any kind of infringement, or somebody wants to check the dates, this has to be, they look into the lab records. That's how you prove that this is your invention or this is your discovery which you made. So make sure that you should do and maintain the lab records over here. With this, friends, I will stop here. This is our certain do's and don'ts for a new lab personnel, or who is never been to a lab or want to go to an international lab. More or less they are the same, and if you follow these things, so it will makes your lab working in that particular setup makes easy. I hope you get benefited from this. If your answer is yes, so please subscribe to my channel, and thank you very much.